Good afternoon, everybody. Thank you for coming today in the middle of the summer. You should be in like some beach resort somewhere, but we're all here still working. I'm Lynn Singer, and some of you I haven't met before, but we're thrilled to have you here and to hear our speaker today. A couple of announcements that just to think about. One is that Autism Speaks, and I don't know if anybody's yet involved with the walk. Last year we had a case, the International Center for Autism Research and Education walked in the annual march. It was in August at that time. It was the day our brand new students were coming on, so it wasn't a good day. But they've changed the date to September now, I believe. This date isn't on here. This is for the kickoff dinner. David, I know you went to the kickoff dinner because David is one of Autism Speaks grantees, really one of the first grantees from Northern Ohio. Do you remember what the date is for the actual walk? I hope anyone here, we'd love to have as many people as possible walking as part of the International Center delegation, so please, we'll send out the information to you so that you'll know. Also, Frank Fexer from PEP, they're usually at this group, but this is the end of the year for them. They're looking for a program director for their PEP Prentice Autism Center, and we'll send that out to you as well. Looking for somebody with a master's degree in education, psychology, or related field to lead their autism center to excellence in operations. So it's something to think about if you know anybody that's interested in that job. And now it is my great honor to be able to introduce to you our speaker today, Dr. Roberto Fernandez Galan, who is an assistant professor in our Department of Neurosciences here, and a Mount Sinai research scholar, as well as an Alfred P. Sloan Research Fellow. And many of you probably saw Roberto's publication that came out about 2 months ago, in which he talked about connectivity and ways to detect autism from brain activity. There's been a great deal of interest in that. In fact, the president of the Hartwell Foundation specifically came and asked to speak with Roberto about this when he was here. And I invited him to give a talk to us today so that we could understand what's happening and what he's doing. So without further ado, Dr. Galan.
Thank you very much. Thank you, Lynn, for the nice introduction. It's a great pleasure for me to have this opportunity to present some of the research going on in my lab to this audience, and I'm hoping to spark some, collaborations from this interaction. So I'm going to be talking about how to detect autism from brain activity. But before I get into the main topic of my talk, I would actually like to give you an overall introduction of the research that we do in my lab. So we have several projects going on, and about 30% of the research that we do is actually experimental. And we do mostly electrophysiology on mice. And 70% of the research that we do is more computational. And by computational, I mean mathematical, theoretical, as well as data analysis, which is going to be the main focus of the talk today. So some of the experimental project that we have in the lab with my student, Pavel Puzarey, is to investigate cortical network excitability and how neuromodulators affected, specifically serotonin. We have found a very interesting link between serotonergic signaling and epileptogenic activity, and we are hoping to publish those results soon. In a collaboration with the Cleveland Clinic, we are investigating alterations of functional brain connectivity in epilepsy. That it's an ongoing project. We are about to submit that for publication. We have some results where we show that based on alterations in functional connectivity, you can predict the outcome of epileptic surgery. So another collaboration that we have with the Department of Medicine at Case is the quantification of cardiorespiratory interactions. And this is mainly with Dr. Thomas Dick, Ted Dick, and also with one of his students, Rishi Dhingra, who is also here in the audience. And we also do some theoretical work on how to simulate activity of ion channels and how they gate ions to the membrane. And the reason why we are very interested in that is because the ion channels are actually random devices, and they generate variability in spiking and also in synaptic release in neurons. So it's like a major source of variability in neuronal networks, and that variability can be positive, but it can be also deleterious, and actually it can be responsible for some diseases like ataxia and cerebellar ataxia. So for the last few years, one big question in my lab has been to investigate the relationship between the structure of a neuronal network and the neuronal activity in that network. Because the structure, the connectivity of the network constrains the activity of the neurons and therefore the information flow in that network. So we have been investigating this from a theoretical perspective and with simulations. A few years ago, shortly before I arrived here, or shortly after actually, I published a work showing that from the activity of a neuronal network, if you are able to actually record it, you can infer a lot of things about the connectivity. You can't figure out the exact connectivity, but you can infer a lot about it. So this study was focusing mostly at the level of microcircuits in the cortex, and specifically in the visual cortex. But then we went on to see if a similar approach could be applied to large-scale brain connectivity, so the kind of neuronal activity that you would record with EEG or MEG. And that's what we did with one of my students, Carl Steinke, who is now a successful engineer in the industry. And we showed that you can reverse engineer a lot of, or many features of the connectivity of a brain just by looking at the traces of activity of the recordings. So one prediction that we had is that in several diseases, and specifically in autism, you would expect the brain to be more homogeneous in several ways that I will be showing to you today. But for that prediction, we needed real data, okay? So these two studies are actually computational, and they try to be as realistic as possible, but obviously they are not the real brain, right? So to investigate that, I teamed up with two collaborators at the University of Toronto who have been investigating MEG in epilepsy and autism for the last few years, and they actually have several publications on the field. And I gave a talk at their university last year, and they told me that they had exactly the data that I was looking for. And as you see from their names, we all speak the same language, which is sometimes advantageous for a collaboration, right? So the question that we had, the questions that I encouraged my collaborators to work with me was, can we detect autism from patterns of brain activity? And the answer is, yes, we can. And that's the reason why I am here today. But basically, in the remaining of my talk, what I want to tell you is how we do that, okay? So I'm going to go through 3 steps. The first one is, I'm going to tell you what are the signals that we actually record. And those signals are MEG, magnetoencephalography, and I will introduce to you what that technique is about. And obviously, we do that with controlled children. We also do that with autistic children. And we look for differences between them. So the second thing is going to be more methodological. I'm going to tell you what do we do with those signals and what kind of information we get from them. One is going to be about interactions between different brain areas, which we refer to as functional connectivity. But one nice feature of the method that I'm going to show you is that in addition to that, we can also infer the inputs that drive the cortex and that are external to it. So it's not, we are going to be able to infer what kind of activity is driving the cortex. So let's go to the methodological part briefly. I'm going to give you a brief introduction to MEG and signal analysis. So the magnetoencephalography basically the technique consists in measuring the magnetic fields generated by the brain. So when the neurons fire, you have like, the currents that flow through the dendrites and so on the circuits, they generate electric fields. That's what you record with EEG signals, with electroencephalography. But they also generate magnetic fields because, that's a physical principle that when you have a current, you generate both electric dipoles and magnetic dipoles. So that's basically what it's sketched here. So these would be like pyramidal cells in the cortex. They are like very well arranged with a given direction, and the flow, the electric currents flowing through those dendrites generate magnetic fields with this orientation. So magnetic fields are much weaker than electric fields, and that's the reason why you have to do those recordings in special chambers, which isolate you from all electromagnetic fields, including the magnetic field of the Earth, which is much stronger than the one that your brain generates. So you have that chamber, and then the magnetic fields are so weak that you need special devices to detect them. And those are made of superconductors, and the superconductors work only at very low temperatures. So you need like a cooling device there. You need like liquid air going through those devices and those sensors so that they can actually detect the magnetic fields. Now, as you see, this setup is much more involved than the one that you have with an EEG. Okay, the EEG is just you put your electrodes in your scalp and you can record it and you don't worry too much about other interference, right? So this is much more involved and it's much more expensive. But the reason why people still do this is because MEG has several advantages relative to EEG. So one advantage is that you have better spatial resolution. And because of that, you can also use many more sensors than the ones that you would use with EEG. For example, with EEG, you have typically 20 electrodes. With MEG, you would have like 150 sensors. So you can accommodate many more sensors over the scalp. One disadvantage of MEG relative to EEG is that you only record the tangential component of the field generated by the neuronal activity. But the good news is that it's less distorted. So what that means basically is that although EEG can capture more signals, if MEG detects them too, those are going to be more reliable than the ones that you would record with EEG. Because the magnetic fields propagate or decay much quicker than electric fields, the MEG is basically sensitive to cortical activity, okay? So the upper layers of the brain, where most of the cognitive function is supposed to be. And another advantage of MEG is that it's reference-free, so you don't need a ground, you don't need a reference electrode or a reference sensor that might be contaminated and contaminate the whole acquisition of the data that you are making. So those are basically the reasons why it's worth investing money in constructing MEG relative to EEG, and that's basically the technology that we have used to record neural activity. Now, once you record activity, what does it look like? So these are real traces recorded from a control child. And there are several features that I would like to point out in this plot. So the first one is that you have like a very stable baseline. Okay, so basically you have fluctuations around a mean, a steady mean. And in fact, you can plot the distribution of values around the mean, and you see that the mean is pretty stable there. But then you have some distribution, and that distribution is telling you that most of the values occur with higher probability around 0, and as you go farther away from the mean, they become less and less probable. So that's a signature for stability, okay? So you have stable activity, you have a stable brain, and it's dominated just by fluctuations. And if you do that for an arbitrary number of channels, as long as you are focusing on resting state activity, so when the brain is not being driven by sensory stimulation, or when you are not paying specific attention to anything, then you're going to have this property. And that's going to be an important feature for what I'm going to tell you later. So basically what we have here is the distribution of amplitudes for each channel. And you see that they fit very well a Gaussian, a normal distribution. Okay, so that tells you that you have like a very stable recording, but the fluctuations are random. Now, the second feature that I would like to point out from these traces is that although they are random, they have like some random structure, they are not independent from each other. For example, you can see here that these fluctuations appear in these two channels, but they are absent in the third channel. So that means that these two areas of the brain from where we are recording these are more cooperative, are more connected. And you can see that in other parts of the signal too. There is like a lot of similarity in the profile of the upstrokes and downstrokes of the signal as opposed to this other one. So some of them are correlated, some of them aren't. And actually you can quantify that. So if you take, at each point in time, you plot the value of this signal versus the value of that signal, what you get is this plot. So for the three signals, you can have like three pair combinations. This is X with Y, this is X with Z, and this is Y with Z. So as you see, the Y, X and Z, or Y and Z are very uncorrelated. You just get like, a cloud of points without any shape. And you have here the correlation values, which are pretty close to 0. As opposed to X and Y, which are highly correlated, and you have like a nice trend there. Okay. So you can do that for any pair of signals. And in particular, you can do that for the data that we collect from the children. And if you do that, so this is the correlated state that I showed you, you can build a matrix where, for each channel, you compare each channel with all the other channels, and you plot the correlation. So this technique is widely used in fMRI studies, and it's fine to use it. And let me show you what are the things that you can do when you build this matrix, which is telling you which pairs, which two areas seem to be correlated with each other. So the first thing that, or one of the things that people typically do is they set a threshold and say, OK, all pairs that are correlated above, let's say, 50% are connected, and I consider them as being functionally coupled, whereas the rest aren't. And the reason why people do this thresholding is because this matrix is basically equivalent to what it's called a graph, which is you can basically plot all the nodes of the network in a circle, like here, and then you can say who is connected to whom. And the reason why people do this analysis is because there is a lot of work done in graph theory that can tell you how random this connectivity is, how many hierarchical structures you have, which are the nodes of the network that are most relevant. For example, in this particular case, what you can see is that just by plotting the connections between all the nodes in the network, not all of them are equivalent. There are some that seem to be receiving many more inputs than others. Okay? Now, we could pursue this, and we did this to some extent, but this approach is not very promising for the data set that I'm going to show you. And the reason why basically this approach didn't allow us to separate between autism and non-autism is because correlations have like several limitations. Okay? Using this approach as functional connectivity has also advantages, which is it's computationally very efficient, and it's applicable to all data sets regardless of the assumption about them. But the caveats are that it's not sensitive to directionality. So you can say whether A and B are connected or not, but you can't tell whether A is driving B or B is driving A. So you don't have information about the direction of the connections. And another thing is that although there are positive and negative correlations, they don't really correspond to excitatory and inhibitory interactions. So it's insensitive to basically who's driving whom, whether an upstroke in A is going to precede always a downstroke in B, and so on. So we knowing these caveats and knowing that this measure was not good enough for us to separate between autistic and non-autistic children, what we did was to try another approach that we had used in our computational studies. And that new approach is based on 3 postulates, okay? And that's where a background in theoretical physics becomes handy. The first postulate is that any complex system around an equilibrium can be described with a universal model that I will describe very quickly in a few minutes. The second postulate is that, as I showed you before from those sample traces, in the resting state, when the brain is not paying attention to anything or it's not being driven by sensory stimulation, the brain is actually in a stable state. So the third postulate is actually a corollary of these 2 postulates, which is that we should be able to describe the neural dynamics of a resting state brain with that universal model. How does that work in practice? So this is a sketch where we have only 3 areas. So let's pretend that our MEG signal has only 3 nodes, three recordings, okay, 3 traces, each corresponding to a different area, area 1, area 2, and area 3. What we record with the MEG is just the output of it, so the activity is basically what we call output, and it's the output from each node, okay? But we know that that universal model that I was talking to you about also considers inputs that we don't know what they are, but it also considers how those nodes interact with each other and how they do that in an asymmetric way. So how node 2 or area 1 is driving area 2 or vice versa, and how area 3 is driving area 2 and vice versa. Okay? Now, basically, we know, I don't want to scare anyone, but this is the most generic model that you can figure out of brain dynamics. Okay? And what I'm going to show you is basically we know the X, we know the output, which is what we record. We also know the rate of change of those outputs. And by fitting our data set to this model, we can retrieve these parameters, the W's. W stands for weight, the fictive synaptic weight between these guys. So basically, those parameters are telling you how they are connected with each other. So by fitting the data to this model, we retrieve how they are connected. And in addition, we get also the inputs that are driving the cortex and that are external to the brain. Okay? So basically, let me try to express this or to reformulate this in another way. This network here is basically the cortex that we can sample, that we can record with MEG, and we know the activity of those areas. But by fitting that activity to this model, we also retrieve the parameters that tells us about the interactions, as well as the inputs that are external to the cortex, but that also contribute to the activity of the nodes in the network. Okay? So one check that you do when you do data fitting to a model is how random are the residuals. If these residuals had some structure, what that means is that your model is not accounting for all the relevant dynamics. So I'm going to show you just like a brief control or reality check that the residuals are actually random, so they are Gaussian, and they are white, so they don't have any temporal structure. Maybe this is a technicality that I don't need to stress, so let me skip it. But the point here is that we have a very good model of the network dynamics in the brain resting state. Well, this is a good validation for that. So the way in which we now define connectivity through those synaptic weights has some advantages and disadvantages as well. So the advantages is that it's computationally efficient. It's sensitive to the directionality of the interactions. So it's going to tell you whether A drives B or vice versa. It can also resolve excitatory and inhibitory interactions. And it allows you to infer the inputs to the cortex, which is something that no other method can do. The disadvantage is that it's a parametric measure. What that means is that it is assuming that your brain is in the resting state. So this kind of approach only will work when you have resting state activity. So when you are not asking the individuals in the MEG to do any specific cognitive task, when they are just resting. Okay. So now that was maybe a little bit too much theory. Let's go to the results. Let's go to the application to autism and how we apply this. So what's the cohort description that we consider here? So we had two groups, 9 autistic children that had been diagnosed at the hospital with Asperger's syndrome, and 10 non-autistic control children. The age was matched. The gender was not matched because, as you may know, autism is much more frequent in boys than girls, and that was, in principle, an initial problem, but I can discuss with you later why this was not a real problem. So the MEG, the grid of sensors, has like this arrangement, okay? So these are the occipital areas of your head, the frontal, the central, temporal, and parietal, okay? So that's our grid of sensors, and basically we are gonna estimate the weights between any pairs in this network. So we are gonna build an array, a matrix, and we are gonna estimate how any two of these sensors are coupled to each other. So when we apply our method, we retrieve those arrays, and now what we want to compare is how different are those arrays. So basically, those connectivity matrices, W, I don't know if everyone can see it, it's also over there. Each of these matrices is gonna be plotted as a point in a high-dimensional space, and we are gonna project it down onto the most relevant projections, which are called principal components, and this is basically what we see. We see some clustering of the connectivity matrices of the control children relative to the autistic children. So what that suggests is that already at this level, we can separate pretty well one group of children from the other. Now what we want to do more in detail is, okay, so those connectivity matrices as a whole seem to be different. Let's now pay attention to what kind of interactions between different parts of the brain are actually different. So for that, what we do is, this is the group of all connectivity matrices for all the children in the control case, and this is for the case of the autistic children. So what we do is we choose a specific combination, let's say one temporal electrode with one occipital electrode. We collect all the values of that pair for all the children, and we compare it with the same for the autistic children. And basically the question is that particular interaction between area 1 and area 2 significantly different in autism relative to control? So that's the question that we are going to ask. Do you have any questions thus far? So when we do that, then we can plot which pairwise interactions are the most the most significantly altered in autism. And what I'm plotting here basically is the change of the connectivity relative to the connectivity in the control case. And then we multiply that by the z-score, by how much significant that alteration is. So you see that there are like many changes in autism relative to control, but actually by far the strongest one, and it's really dominant, it's this interaction between the parietal zone and the frontal zone. What we have is massive increased excitation along between these two areas, okay? Between that frontal area and that one. And that's very significant. That's the reason why these values are very high. So there are some other changes that we haven't investigated in that detail, just because we focused on the largest one. Okay. In the literature, there are several studies that have shown alterations in white matter density, and some other things, like, for example, in particular, my collaborators had shown before that there is increased synchrony of fluctuations in this part of the brain, the parietal one, parietal occipital, and frontal. So in a sense, this is consistent with what some other people have seen. Now, to be honest with you, I was a little bit surprised about this connection because, I don't know any pathway in the brain that matches that directionality. I was expecting something in the other direction because, if you think about, for example, modulatory alterations, then you would expect the changes to be from the front side to the back side, right? Like, serotonin, dopamine, whatever goes from in this direction, right, backwards. But when Takao Henge was here 2 weeks ago, I showed him the data, and he said that that's exactly what he expected. And the reason for that is his reasoning was the following. He said, autism is a neurodevelopmental disorder, right? And what they have seen in mice is that the development of the brain first occurs from the back to the front, and then at some point it continues from the front to the back. So he was actually happy to see this, and until then I didn't have an explanation of why this particular direction might be relevant. Okay. Now, I told you before that in addition to investigating alterations in connectivity, we can also infer the inputs to the brain, to the cortex. So what we did was to plot the input distribution throughout the cortex and see if we could find any differences. And now my question to you is, can you see any differences between control, which is the upper two layers, and the autistic children, which are the bottom ones?
The autistic inputs are more monotonous. They're more uniform.
Someone disagrees? It's pretty obvious, although it's not perfect. I mean, there are some cases that are a little bit, you could be uncertain about where you would classify them. But the trend is pretty clear. In autism, the pattern of inputs is much more homogeneous. And you can quantify that homogeneity. So in particular, you can quantify the spatial complexity. And you see that in the control case, that complexity is much higher. So you have like a more homogeneous, less complex pattern of inputs to the cortex in the autistic children. And that's highly significant. Okay, so now in the last few minutes, the last few slides, I'm going to show you something that is not published yet. What I have shown you thus far was already published in this paper that Lynn was referring to in PLOS ONE. And now this is new results on a follow-up paper. So autism has been defined on many occasions, but since the early days as withdrawal into one's inner world. Okay? And the first person who used that term was actually Eugene Bloyer, who first reported autism, although he referred to it as the children's version of schizophrenia. Okay, so he referred to it as the schizophrenia of the children, because he noticed that, in schizophrenia, when the disease is developed, you have similar symptoms in the sense that you lose interactions with the external world and things like that. So he referred to it as withdrawal into one's inner world. But then the same definition, although refined, was used by Hans Asperger, who gave name to the Asperger syndrome, and Leo Kanner, who defined autism the way that we would understand it today. Okay, so he departed a little bit from the original definition, but all of them had in mind this idea of describing autism as withdrawal into one's inner world. So now the question is, can we quantify withdrawal into self? Is it possible to do that from data? I mean, is there a quantitative version of that statement that comes from cognitive science? And that's what we wanted to try. So basically, if you think in a more abstract way, what I have shown you today is that you have the brain as a black box. And you can focus on the black box itself. You can focus on how things interact within the black box, which is the connectivity. But you can also zoom out and think of this as some transformation between a set of inputs and a set of outputs. Okay, so you can think more like as an engineer. And I showed you also that that's what we measure, and that's what we can infer from the data. So if you go back and think as an engineer, the question is, what's the information gain of that process? I mean, how much of the input is actually transformed, and how much of the output actually explains the input, or how much variability of the output is actually new? So this can be quantified very accurately. The information gain is defined as the entropy of the output relative to the input. Let me explain that. What we are measuring now, what we are going to be measuring is how much variability of the output, both in time and space, can be accounted for by the variability of the inputs. Variability in terms of time, the fluctuations, as well as in terms of space, the spatial variability that I just showed you. Okay? So that can be rigorously quantified. And that's what we are trying to do. So obviously, if the brain here was not doing anything, you would expect the information gain to be 0, because what you put in is what you get out. Now, if you have some kind of complicated transformation, then you're going to have a higher information gain. So again, I don't mean to scare anyone, but basically you can compute that rigorously, and you can't do that in the general case, but you can do it if you focus on the resting state. In the resting state, this expression is very well defined, and actually there is an analytical expression that you can calculate just from the covariance of the inputs and the covariance of the outputs. You can exactly quantify how much information of the outputs is new. And if you do that, this is what you see. So on the left, you have the information gain for each of the individuals in control. And on the right, you have the information gain for each of the individuals in the autistic case. And as you see, there is quite a bit of an overlap. There is a lot of overlap. But the medians are significantly different. And in fact, the change of the medians is 42%. So what that means is that the autistic brains, on average, create more information from the inputs that they receive. And we like that interpretation because it coincides with the hypothesis or the description that autism is actually the withdrawal into one's inner world. Why? Because you don't need external stimulation to actually have some complex brain activity. So it's, in a sense, we think that this is consistent really with a brain that is so occupied with itself that it's not much affected by what it receives from the outer world. So in summary, we have applied a universal model to determine functional connectivity of the cortex as well as its inputs. Two advantages of this measure is that the functional connectivity that you get is directional, so you can resolve asymmetric interactions between any two parts of the brain. It's sensitive to both excitation and inhibition, but in addition to that, you can resolve the inputs to the cortex. And one thing that we have shown in terms of connectivity is that there is a massive increase of excitation between parietal areas and frontal areas. And finally, what we have shown is that brains from autistic children generate more information in the absence of stimulation, which is consistent with the description of autism as withdrawal into self. Okay. And thanks a lot for your attention. I should mention that we didn't have any specific funding for this project, but we are thriving to get more. Okay. So thanks a lot, and please contact me if you're interested in a collaboration in this field. Thank you.
Any questions? One, do you perceive that this, one of the goals in autism research is to get a diagnostic tool that could early on identify so that kids could get into intervention. Do you see this as something that could lead to that, or could you?
That's where we would like to go. That's
something eventually that.
Exactly. That's where we would like to go, and for that we need more patients, obviously. So I think we have like a very promising biomarker for at least for a specific form of autism, but we would like to verify this with a larger cohort of patients, but yeah, we are very interested in that.
Yes.
So, two questions. So, give us some context here. Has this type of computational analysis been performed with other methodology, or is this?
Right. So the methodology that other people apply, and sorry if I didn't make that clear. What people do is this analysis that I mentioned before, this correlation analysis with the graph theory and so on. But if you apply the same approach to our data set, you wouldn't get the same level of separation between autistic children and non-autistic children. Okay, so the gold standard, if you will, of current analysis wouldn't be able to resolve the differences that we have observed.
So clearly, the clinical spectrum presentation of these patients is broad. So were these patients selected so that their clinical picture was the same, or was this? And would you expect that this outcome to change?
Well, that's exactly what we would like to know. So, we would like to know if, with a larger cohort of patients that is more diverse, we would systematically see the same changes. So one thing that I should say is that the patients themselves, I never interacted with them, but they were very heterogeneous, the autistic children. The people who interacted with them told me that. So it's surprising in a sense that in spite of having like a different phenotype, so to speak, they are still categorized in the same, they are cast in the same group. So, I think that's an interesting feature of the method. But, I think, we need a larger data set, obviously, to check if that can become a biomarker, But we think, based on this, there are some features that are very consistent, and in spite of that variability in the phenotype.
And could you talk into the microphone, because it's hard for people to hear the questions, I think.
Can you hear me? Is this one on?
If it's got a green light, it should be working.
It's just red.
Oh, forget it.
Red or off.
Shout.
Yeah.
Okay. This is really interesting. I have a ton of questions, but I'll stick with one for now. In terms of the sensitivity and specificity of the approach, when you looked at the actual individual brain images, where you mapped the actual strength of the effect.
Yeah.
In the control group, you saw some kids who looked exactly like the ASD group. And I'm wondering, whether you actually looked more closely at those cases, if you could,
So actually, we did a specificity and sensitivity analysis. We reported that in the paper. Perhaps I should have shown it. We did it. Okay. I decided to show the results that are more visual. Okay, that can be, but we also have that analysis, and I can tell you that the accuracy is 94% in the classification. But I can also give you the sensitivity and the specificity that I don't remember. It was lower than the accuracy. It was 86 and 84, I think. We did that. Yes. Okay.
So, Roberto, there's been a lot of work with fMRI on functional connectivity in autism.
Mm-hmm.
And the bulk of that work has led to the conclusion that there is hypoconnectivity, at least in certain networks.
Mm-hmm.
Can you tell us how your results, either how they relate to that body of evidence?
Right. So we don't see that with our data set. Now I can tell you, so the reason why I explained this is because I wanted to give an introduction of what people do with fMRI these days, okay? fMRI, it's good, but it has two disadvantages. One is that it's very slow, okay? Neural dynamics is much faster than that. And some of the features that we see, some of the fluctuations that we see with MEG, you wouldn't see them with fMRI. But the other one is that it's not completely clear what the signal is. Still, you can do some functional connectivity analysis like the one that I showed here. If you try to replicate the same technique with these graphs and so on, you don't see hypoconnectivity, and on the top of that, you cannot separate both groups. So the way I answer your question is that with different techniques, you see different things. The connectivity is not the same, right? I mean, what you call functional connectivity when you are using EEG or MEG is not the functional connectivity that you record with fMRI, right? fMRI is more coupled to the vascular system, right, and to the oxygenation of it, whereas MEG and EEG are more focused on neuronal activity proper, right? So I think that we have to be cautious because even when we refer to the same thing, because what we are trying to do is really, we are trying to understand how things are connected, the term functional is key. So functional means that it basically depends on the technique that you use. Now, if you tell me why there is hypoconnectivity with fMRI, it could be because the vascularization of the brain is different. It may be totally unrelated to white versus gray matter in the brain, right? That's one possible explanation. Diffusive tensor imaging has shown that there is like more white connectivity, although it's a little bit controversial, but many people have seen that. And that agrees more with what we see. Because if you have more promiscuous connections between neurons, then you would expect to see more homogeneous input patterns like the ones that we see. So I think for us, it's easier to reconcile the findings that people have obtained with diffusive tensor imaging rather than with fMRI, right?
This is really magnificent, fascinating, and the way I understand the way you explain MEG is a measure of like the global connectivity more at like the superficial cortical layers. But how sensitive would it be to measure like deeper cortical layers or like deeper seated brain nuclei where there are changes in local circuitry there that are perturbed and wouldn't be picked up, but can affect diseases like obviously ASD or other diseases, epilepsy.
Right. So I think MEG detects to some extent deeper layers, okay, but you don't see everything. So that's obviously a limitation of the method. One good feature though is that because it detects more the tangential component of the the magnetic field, you detect activity that it's in the sulci of the brain, in the grooves, rather than on the gyri. And I think that gives you some sense of depth, but obviously you are not sampling really from deeper areas of the brain. I think the integration of techniques is the ultimate way to go, which is what people do when they try to find the location of an epileptic seizure, for example. But what I have learned from my collaboration with the people at the clinic is that when you apply different methods, sometimes you get contradictory answers, right? So for example, fMRI tells you that the seizure is at one point, MEG tells you that it's not, that it's on the other side of the brain. So, I mean, there is room for improvement in those techniques. MEG has its limitations, okay? One is what you are pointing out, I mean, how deep you can go. And, that's an important thing to keep in mind.
That's like just a follow-up, and that kind of leads to Gary's question or the one before. So could it be, since you are doing such a global measure, could it be the reason you get a homogeneous kind of readout in a set of ASD? I mean, if you go to a more like, Fragile X or Rett for that matter, would you get a different signature of the brain altogether in those kids?
I don't know.
How generalized can this be, coming back to Gary's question?
I don't know. So in principle, you can apply the same method, but obviously, I can't predict, right? I mean, it would be interesting to do exactly that, to see, is this specific to Asperger's, or, it's more general than that. And, I don't know, but I'm happy to test that. Yes.
Along with that, are you able to apply behavioral correlates to your model so that you can look at, as you start to expand? My assumption is that these kids have Asperger's, so they're all within the normative range of cognition, but as you expand in the ASD data set, cognition is important, but also if you think about, sort of withdrawing into your inner self, perseverative thoughts vary quite a bit between people with ASD, so being able to apply a behavioral variable would be helpful.
Right. So let me tell you something about the technical challenge of doing that. When you ask the children to do some tasks, you are pulling them away from the brain's resting state, right?
Not during standing.
Oh, okay.
So you should have behavioral and demographic data on these individuals. Can you pull that information?
We didn't have access to that information. We only have, we have the IQ, interestingly, okay? But here's the thing. When considered as a group, the IQ of the Asperger's and the control children was the same.
So, but is it possible, using your more complex model, to be able to add one of those variables to look at distinctions within the autism group on perseverative interests, cognition, gender?
It would be possible in the sense that, one thing that would be possible to do is some sort of multivariate analysis where you try to find correlations between some of the features that we see altered, and then some behavioral, facts or something, behavioral features, right? So in principle, it's possible to do. Unfortunately, we don't have that. But if we as a group, also, I'm talking now more as a group coordinated here, if that information was available, it would be certainly possible to try to look for those relationships. And that would be really interesting.
Do you have some independent measure of what constitutes a resting state? Because just thinking about the population, a group of people that tend to be a little bit anxious with a very large cone and full room, they may not be at the same resting state that somebody else is.
You are right about that. So that's the reason why we use like a more empirical definition, which is how stable the mean activity is. Let me go back to. So if you have a stimulated brain, what you're going to have is the baseline is going to change quite a bit, right? Like if you are focusing on something, or you turn your head or something, some areas are going to violate that assumption that the mean is not steady. Okay, so I mean, at least we have some empirical measure for that. But in terms of behavior, really, we don't.
But if you're just recording from superficial cortex, it may not be sensitive to areas where you do have differences based on state.
Right, but I mean, for our model to work, it doesn't matter. So maybe I emphasize too much the necessity or the limitation of being in the resting state. What I really mean by that, it's not that much cognitive. is more in the sense that you need the signals to be stable, okay? So it can be that you might have some, thalamic, actually, obviously the thalamus, we think, by the way, that the inputs to the cortex that we estimate are thalamic, right? I mean, based on, so I mean, even if you don't see it directly, indirectly, you feel the thalamus in the signals, right? But that's the point, that, they, as long as you have some steady signals, you can apply the method.
So you focus on this one connection, if you will, that seemed to be much stronger in the ASD group. But when you look at connectivity globally, in the ASD population versus the control, how does it compare?
So these, you're talking about this plot on the right? So this encompasses, basically when we do this plot, we are considering all possible alterations, including the one that I emphasized later. So this is like a more global analysis of the network that doesn't focus on which particular pairwise interactions are altered, but instead it focuses on the networks as a whole and how well you can segregate them. Every pair that it's altered contributes to that difference.
So the fact that the ASD group is to the left of the control group, does that mean more or less connectivity?
You can't tell because this is a projection of the actual space. So here we have 140 sensors, and here we have 140 sensors. So when you plot this as a point, you have 140 squared dimensions, right? You can't visualize that, obviously. So what you do is you project it down to the most relevant dimensions, and then what's right, what's left, what's top, what's bottom, you can't tell a priori.
But how do we interpret that graph?
That graph is just a visualization of that high dimensional space that allows you to see that, there is some clustering of the phenotypes.
It doesn't tell us if there's more or less connectivity in one group versus the other.
Correct. That's correct. We ran several analyses that I didn't show you. In terms of activity, you have the same level of activity. So it's not that one brain is less active, the other one is more active. We did also what it's called a linear stability analysis. It's basically a test of how epileptic your brain is. They all are the same, and they are very stable brains, not pro-epileptic. And in terms of connectivity, besides what I showed you, I cannot tell you, I can't be much more specific than that. I mean, I can show you all the arrows that I was mentioning here, right? I mean, there are many changes that are highly significant, but by far, I tend to focus on the things that pop up the most, right? And that's really the guy who is not only the largest relative change, but in addition to that, the most significant one. But as you see, there are like many other things going on, and they tend to be all excitatory. Okay, so if you compute the average of those things, it's more excitatory than inhibitory. And in MECP2 mice, for example, people have shown actually an increased excitation, and actually you guys have done the same analysis. So this is consistent at another level with increased excitation in the brain.
No, in MECP2 you see reduced cortical activation. There's less excitation.
Cortical, but
there's less excitation in cortical surface.
Oh, okay. So then that's not what we see at this level.
So then coming back to David's question, when you measured the overall activity, did you measure the variance or the difference in their beta or in their theta?
You're talking about frequency bands now?
Yeah. About when you said you did the epileptic activity in them, and they had similar activity all over.
Right. No. So we did the analysis another way, and I can give you the details of that. But we also did a spectral analysis analysis of the brain activity to see if there are differences in the frequency content. There is not. Okay, so in the resting state, you don't see a lot of rhythms. The one that was very prominent in both groups, in the control and the autistic children, was the alpha rhythm, but that's because they had their eyes open. So I mean, they were not focusing on anything special, but they had their eyes open. And it was not significantly different between both groups. And we didn't see gamma rhythm or beta rhythm, which is consistent with the fact that they are really resting. Right.
So I saw one last question, and then you can talk to Roberto personally afterwards if you wish. Lynn.
You did a very nice job of explaining the pros and cons of MEG versus EEG, but if you had EEG data on these same children, would you be able to apply the same analysis and show the same results?
Okay. To the first question, yes, this analysis can be done with EEG perfectly. Now, to the second question, that goes back to David's question. Functional connectivity depends on what you are measuring, right? Obviously, there is much more correlation between the magnetic fields and the electric fields than magnetic fields and fMRI, which is the oxygenation of the blood. And that's because the electromagnetic fields themselves are coupled. So I mean, right? They propagate differently, but they are generated, coupled in a coupled way. So the only things to keep in mind is that if you see a different pattern of connectivity, that might be because with EEG, two things. You measure more activity. It's more sensitive, but it's less precise. Okay? So those would be the things to take into account. It can be applied, and you could see more things, but with less reliability. I actually was more interested in the EEG because I have a better understanding, of things. But that's what we had, and you get the additional perks that you have, like more spatial resolution, which is also good.
But EEG is a lot easier.
Cheaper, easier, yeah, exactly. Yeah.
Cheap is good for researchers these days.
Right.
Well, thank you, Roberto. That was with the patient registry and the biorepository that's being developed as we speak, it seems like, rather than going to Toronto, you should be able to get some of your data here, working with everybody in this room. So
That'd be great.
We're pleased to see what you're doing. Thanks. Come back again.